So pleased to welcome Jessica Wapner tonight to discuss her book, The Philadelphia Chromosome, A Mutant Gene and the Quest to Cure Cancer at the Genetic Level. The book tells two stories. The first traces the groundbreaking research of genetics, from knowing nothing about genes to understanding how they can cause cancer. And the second is a thriller. Will this drug, Gleevec, am I pronouncing that right? Gleevec, a drug that could make a fatal cancer treatable with a single daily pill, would it be produced? I liked what David Quammen, who's another great science writer, he had to say about the book. He says, Jessica has done two kinds of hard work gracefully. The hard work of understanding cancer genetics and the hard work of rendering that subject into human narrative, lucid explanation, and metaphor. The Philadelphia Chromosome is not just an urgently useful book, it's also an elegant one, put together like a Swiss watch. Jessica's a freelance writer whose work has appeared in Scientific American, Slate, and the New York Times, among many other places. Please join me in welcoming her to Politics and Prose.
I first started researching this story. I first heard about the Philadelphia Chromosome around the year 2000. I was working at a medical journal, and that was close to the time when the drug Gleevec, that is at the heart of this book, was approved. I didn't know much about it. I would hear these words, the Philadelphia Chromosome. I mean, it's such a great name for a genetic mutation. I mean, they should all be named something so captivating, so memorable. I knew it had to do with this rare leukemia, but I didn't know what. And a few years later, as I became a full-time science writer, I started asking some more questions about it. I wanted to know a little bit more. And it all began with asking, where had this name come from? The Philadelphia Chromosome was named after the city where it had been discovered in 1959 by two scientists, David Hungerford and Peter Knoll, who were simply looking through a microscope at samples of blood cells from people with leukemia and spotted this abnormally small chromosome. It's on the cover of the book. It was completely unlikely that they would even notice it. It was a simple microscope they were looking through. Just chromosomes at that time just were like ink blots. And for David Hungerford to have noticed that one of them was too small, there weren't many people alive at that time who could have seen that. So that was interesting to me. The Philadelphia Chromosome, how somebody had discovered it. And at the time, I thought, maybe a magazine article. But then I began asking more and more people about it. I ended up interviewing more than 30 people in the end about what was the story behind the Philadelphia Chromosome, how had science led to the unraveling of the connection between this chromosome and this fatal cancer, and what had happened after that, when that picture became clear. And there were so many twists and turns in the story. So, for example, the Philadelphia chromosome is this genetic mutation in which pieces of two chromosomes, chromosome 22 and chromosome 9, swap places. That's it. It's just a tiny little change. It can't be predicted and it can't be prevented. And it is diagnosed in about 5,000 people in the United States each year. That mutation, that mutant gene, encodes a mutant protein. And that mutant protein, which is called a kinase, the kind of protein it is called a kinase, is what triggers the production of white blood cells. Okay, so normally white blood cell production, we have a red light and a green light. Our bodies do not always need to be making white blood cells all the time. In people with the Philadelphia chromosome, that mutant protein, the mutant kinase, is always giving that signal to produce white blood cells. It's green light all the time. So I'm asking somebody, well, how did kinases get connected to cancer? Where did that all start? And the first person I spoke with led me back to a man named Ray Erickson, who had been studying kinases in Colorado. He wanted to uncover the protein encoded by a virus that was causing cancer in chickens. So I'm talking to Ray Erickson, well, where did that come from? And he said, this Sark gene that I was studying which led to the first discovery that kinases are involved in cancer development, in some cancer development, that had come from the Rous sarcoma virus, something named the Rous sarcoma virus. So I asked him, well, who's Rous and what was his sarcoma virus? And so what I found out was that actually went all the way back to 1910 to a man named Peyton Rous, who had been studying at Rockefeller University, which is in Manhattan. And one day, about a year after he started working there, this farmer arrives in his laboratory carrying a chicken. And the chicken has a tumor sticking out of its belly. And she says to him, "Can you operate?" The tumor that he took out of the chicken ended up, through decades of research, leading to scientists understanding not only the role of kinases in some cancers, but also the genetic underpinnings of cancer. The fact that cancer can have its root in our own genes. And that was just one example of all of the twists and turns. I mean, can you imagine this happening today? A farmer walking into a university carrying her chicken? The paperwork alone would just send her away. So as I started understanding, pulling on these threads and more, there was the Abelson virus, which was discovered at the NIH right here. I thought this is way more than a magazine article. This has to be a book. This has to be written down. Many of the researchers involved in this work, this was decades ago, and I knew that the time to catch their story was finite, and I wanted to make sure that it would be recorded, and recorded correctly. So that was enough to set me on this trail of this has to be a book. But then the story takes a whole different turn, as Sarah spoke about, where it becomes this mystery of how is this drug going to get made. And I'll tell you what I mean by that. By about 1984, 1985, scientists had fully pictured out, fully unraveled how this mutant chromosome leads to CML. As I explained, this swap encodes a mutant protein, right? Normally the protein helps trigger production of white blood cells. In its mutated state, it's always got this green light signal, and the body gets filled up with way too many white blood cells. So when that picture became clear, people are starting to think, well, wait a minute, can we make a drug to target that? Can we just create some chemical that will just throw a wrench into the clockwork and stop the cancer at its root cause? That had never been attempted before. That was an entirely new way of thinking about drugs for cancer, right? Up until that time, the prevalent medication for cancer was chemotherapy, which, of course, chemo means poison. It's poison therapy, where you're trying to kill the cancer cells before harming the person beyond what they can tolerate. This was something different. We know the underlying mechanism. We know why this leukemia is happening. Can we make a chemical to get inside the cell and block it? By 1993, that chemical had been made. A company named Ciba-Geigy in Switzerland had started a program in the mid-1980s to create what is called a kinase inhibitor, a kinase stopper, okay? They sent a handful of compounds to Brian Drucker, who I talk about in the beginning, who was researching in Portland. He has an amazing story I'll read about in the book. He was researching in Portland. They sent him 5 chemicals, and they didn't tell him which one was the one they thought would work, right? Obviously, they're not going to tell him. So he tested out on CML cells, chronic myeloid leukemia cells, and he sees right away, one of them is working. It is stopping the cells in the test tube, they're dead. And when he reported back to the company, this was the one that was their lead candidate, right? So So there was a match. There was something that they had succeeded. All signs were pointing to success. Now, of course, that was in a laboratory. There's a long way to go before you get to giving a drug to people. But anybody who sees a compound, an experimental compound, killing cancer cells in a test tube in a laboratory, knows the next step, which is animal studies. Get that as fast as you can into animals. Because if we have a drug here that could help patients, we have to get it to them as soon as possible. Because we're a drug company, we're a business, and that's what we do. And also because at that time, in 1993, there were 2 treatments for CML. One of them was bone marrow transplantation, which is actually still the only curative treatment. But many patients, most patients, were not candidates for a bone marrow transplantation. And even those who were, only about half of the people survive it. It's just such a harsh thing to go through. It takes about a year to recover from. It costs about $1 million. And a lot of times people relapse from it anyway. So the compound was killing cells in the laboratory, and Brian Drucker is in Portland waiting for a word from Siba Geigy in Switzerland about when the animal tests are going to begin. And he hears nothing. Instead of everything plowing full speed ahead, everything ground to a halt. Just nothing. So he's telling me this story. I spent 3 days with him in Portland last year telling me this, and I spoke with people. When I heard this, right, that things had ground to a halt, that this drug, which seemed so promising, was not following this well-worn path of drug development, I wanted to know why. What happened? I want, you know how it is. There's a problem, you want to know every corner, every detail of the story. And I spoke with people inside the drug company. I spoke with patients. I spoke with doctors to find out what had gone on. Now, there were 2 things that had gone on, it turns out. First, this was risky, right? I said this was an entirely new way of treating cancer. It had never been done before. So even though the chemical was so obviously killing cells in the laboratory, there was still no knowing what was going to happen when you put that drug into animals or into people. And the shadow of thalidomide, Jörg Zimmermann, who is the man in Switzerland who actually created the chemical, he told me, you have to understand, the shadow of thalidomide was hanging over the pharmaceutical industry, and it still does. This was a drug that was supposed to help women through their pregnancy and ended up deforming their babies. We've all seen the pictures. Nobody wanted another disaster like that on their hands. And so Jörg Zimmermann has explained to me, he says, look, you can look at the time it took for us to make this drug and say, why did it take so long? But if we had gone faster and a disaster like that had happened, you would have said, how could you? Why did you take the risk? So they were being extremely careful, because if the drug inhibited more than one kinase, that was going to be one sick patient. They finally started the animal studies in the mid-90s. And in fact, there were problems in the animal studies. There were a few problems that you'll read about in the book. And at one point, so the drug was given to mice, rats, dogs, monkeys, rabbits, and cells from other animals, including hamsters. At one point, dogs that were given an extremely high dose of the drug, around 600 milligrams, not extremely high, but that was pretty high for an animal test, started developing liver problems. And the animal studies had already been going on for about a year at this point, maybe more, and these dogs start having liver failure. And one of the toxicologists at the company says to the man who had led the development of the drug in the company. 2 different teams. It's all bureaucracy and fights and things. And what the toxicologist says to this other man, "Not over my dead body will this compound get into man," when he saw the liver failure. Brian Drucker in Portland is saying, "You're nuts. You have an ethical responsibility to get this drug to patients. This might help people that have a fatal disease. You have to try it out. We are doctors, and we know the signs of liver failure. First of all, a clinical trial does not start out at 600 milligrams. A clinical trial starts out at 25 milligrams. And if we see signs of liver failure or liver problems at all, we're going to stop the drug. That's what we do as doctors." So there was this whole aspect of the risk, but that wasn't the only part of the story. And there was a whole other drama that just, I again felt this had to be written as a book, because alongside the risk was also the money. So you remember I told you that CML is a rare disease, right? It's diagnosed in about 5,000 people per year in the US. To compare that, to give you a sense of numbers, breast cancer is diagnosed in about 230,000 people per year in the US. Cancer of all kinds in the US is diagnosed in about 1.6 million people. Okay, so 1.6 million, 5,000. So 5,000 is the patient population per year. In pharmaceutical lingo, that's the market. Okay, so the market for the drug was too small to guarantee a return on its investment. They could not see how was this drug, which was going to cost millions and millions of dollars to make, how were they ever going to get their money back on it? It was, I told you, 1993 was when Brian Drucker had started testing the compounds on cells. In 1998, so 5 years later, a process that should have taken about 2.5 years, about 5 years later, finally the clinical trial started.
After reading the book, I wondered why there isn't, because when the issue came down to, do we make a profit? So therefore, do we go ahead and develop this drug? When the numbers are so small, yet you're on the cusp of being able to eradicate or at least control, because you never eradicate. From what I understand, the drug just controls. It never eradicates. Why we do not have processes involved so that the drug company and NIH, which is a federal sponsored, our taxes, so on and so forth, are not involved in stepping in on that level. I understand the breast cancer, there's enough that they're going to go, and they are, full tilt boogie to try to, because they know there's huge money to be made there. Yeah. But in this case, what I don't understand or what I don't know is in place is the idea on this thing where a drug company goes, we can do this, but it's not going to be profitable. That it seems to me there's the place that NIH and...
Yeah, well, I can respond a little about that. I mean, one of the reasons why it was possible for Novartis, they were Novartis by then, to get the drug into clinical trials was the Orphan Drug Act, which was established in 1983 for this exact problem. Because for a long time, rare diseases were just, there's no rare diseases. We don't have to make medicines for those because of this problem of,
profit over health.
This is for-profit healthcare, right? Correct. So how do we do this? We have on the one hand this ethical imperative to help all illness, but on the other hand, we live in a world where drugs are made by companies. So, and they can't be expected to just do it from the good of their heart, as much as some people would like to, right? It's not to say that everyone inside a drug company is just, with dollar signs in their eyes all the time. So the Orphan Drug Act gave several incentives to help, which included tax breaks on clinical trial costs, and 7 years of market exclusivity when it's first started. I think the number is a little different now, which meant there would be no competition for a very long time. And these were incentives to help. Since that act was put in place, before that act, I think there were about 10 or 15 drugs for rare diseases. Now there's about 250 drugs of this kind. And rare diseases are, suddenly everyone is saying, oh, rare diseases. Let's make drugs for rare diseases. So there's actually a lot more attention being paid to them now than there was back in the day of when a lot of this story is being told. I told you the drug was approved in 2001. It was the fastest approval ever. By 2011, Gleevec sales had totaled about $27 billion. So they had definitely made their money back because the drug works so well and because you have to take a pill once a day and it lets you live a normal, healthy life for the most part. It works financially, which nobody could have predicted. As far as the patent, the story with that is that in India, drug patents are not, before 1995, are not recognized. So when Novartis had originally patented the Gleevec compound in 93, and they went to repatent it in 95 as a slightly different formula, and the Indian government said that they did not recognize it as a unique drug. That happens a lot in our country. It's something called evergreening, where a drug company will change the formula ever so slightly to extend the patent. So what happened in India is that 1995 patent application was rejected, essentially. So generic Gleevec is made there for about, I forget how much each dose costs, but it's a far cry from the current price tag in the US, which is $100,000 per year for second and third generation versions of the drug.
And I'm not a doctor, so I have two simple questions. First, are you born with this Philadelphia chromosome, or can it strike everybody anytime? And the second question is about Gleevec. I guess it doesn't cure; it doesn't just stop the disease. And if you don't take, you have to take it till the end. And what is the life expectancy of a CML patient?
Right. Yeah. So I can respond to all of that. Great. So yes, the Philadelphia chromosome is what's called a spontaneous mutation. It's not inherited. So unlike BRCA1 or other that we hear about, that's the breast cancer gene I was mentioning earlier, the Philadelphia chromosome arises spontaneously. We get many genetic mutations in our lives that arise spontaneously and disappear that don't ever cause any problems, so we never know about them. This one happens to cause leukemia, and it can't be predicted, and it can't be prevented, and it can't be passed on. And yes, that's right. So this pill is something that a person has to take every day. And when people stop taking the drug, the disease comes back. Because, as I said, it's because of how it works. Because it's blocking the kinase. So if you stop blocking the kinase, boom, 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 you're going to get those white blood cells again. There have been some studies of people trying out stopping the drug. I mean, I think there's people who, not on a study, are like, just feel so good and have been fine for so long, they do stop taking the drug. But that's, minimal people. And they've done some studies. In fact, someone came up to me the other night at Fox Chase and said he had been on a study in 2010 of people who were stopping the drug to see what would happen. And 6 months later, his disease started returning. The most current survival data are 10 years. So the clinical trial that started in 98, so there's people now, I don't think there's data published past 10 years yet, but there's the most recent survival data were 68%. So 68% of people who'd been taking this drug for 10 years were still alive. Before 2001, the 10-year survival rate for CML was 0. So 68% to 0. And the 5-year survival rate, of course, is even higher. So the drug company did the right thing in the end. But I felt this story showed us so much about what goes on behind the scenes. I want to know, I want to know everything. If I'm going to think about healthcare, if I'm going to think about cancer and how to make the situation better where cancer is concerned. I felt this story just needed to be told, needed to be written down. Because that wasn't the end of the struggle, because the clinical trial was started, and as you'll read about in the book, the response rates were unprecedented. Unprecedented. So all of these fears of the risk, there was no risk. At one point in the phase 1, which is a trial in phase 1, nobody is expecting to see a response. The purpose of the earliest clinical trial is just to test for the safety of the drug. A few months into the study, every single patient is responding. A 100% response rate. I mean, this is as close to a miracle as it gets. There were still delays with the drug. And there's a moment where the patients are instrumental in rescuing the drug. There was a woman I spoke with, her name is Susan Mcnamara. You'll read about her amazing story. She was so sick, she was living between her bed and her computer, parked 2 feet away. And she heard about this drug. It was incredible timing that internet chat rooms happened to rise up at the same time that this drug was being made. She heard about this drug. She contacted Brian Drucker to get on the trial, and he said, I don't know what's happening. This was between phase 1 and phase 2. Phase 2 is expanded, so there's a lot more people. Phase 1 trials are tiny. In phase 2, a lot more people can get access to the drug. But there's delays again. And Brian Drucker is telling her, I'm sorry. I'll put your name on a waiting list. But his waiting list was at least 100 patients long. And he had to pick and choose who he thought was the sickest to see who was going to get when the next dose became available. And she said to me, there was no way that there was a drug that existed that might help me that I was not going to get. And she wrote a petition to the company, and a few months later, the drug had become widely available. And Brian Drucker had fought tooth and nail, 100-hour work weeks for years, a marriage that fell apart, another marriage that bloomed. You read about his lovely love story in the book. And CML patients around the world credit him for saving their lives. To me, this story, it brings us to our current moment in cancer research. So it's a history, it's a medical history that explains how we got to this moment, right? How it is that Angelina Jolie is using genetic information to make extremely important life decisions. How any of us are using genetic information, why any of us would think about that. This story is the origin story for that. And alongside the science and the medicine, which is so compelling and so fascinating, there was this incredible human story of a miracle drug and the miraculous lengths people will go to make sure that something that they believe in, that something that they know is possible, gets a chance in this world. Which is why I wanted to write the book. So, thank you.